Today we're going to be talking about enzymes. And by the end of today's lesson, you should be able to be familiar with the following learning targets. A catalyst is any substance that speeds up chemical reactions. And we already know that a chemical reaction happens when you are trying to form compounds or molecules. So when you have two or more atoms that are combining with a chemical bond and forming something new, forming a new substance, forming a new molecule or compound, this is known as a chemical reaction. Now, generally, chemical reactions happen too slowly. And in order to speed things up, scientists can use catalysts. So catalysts can be added during an experiment in order to speed up chemical reactions whenever an experiment is being conducted. However, we also have certain types of catalysts that basically are found in living things. They are found in our bodies. And these catalysts are known as enzymes. So enzymes are catalysts that are found in living things, or in other words, enzymes are biological catalysts. Now, enzymes are extremely important for maintaining homeostasis, and they are needed for almost all processes that happen in our bodies. They are needed to keep us alive. And this is because without enzymes, all the processes that are happening in our bodies would take place at a very slow rate, and that will not be sustainable for us to stay alive. Now, most enzymes that are found in our bodies are considered proteins. Okay, so we're going to talk about carbon-based molecules, and basically enzymes are a specific kind of carbon-based molecule known as proteins. So all enzymes have a very specific structure, and their structure is very related to their function. Or their structure, the way that they look like, it helps them perform their job. You'll notice that an enzyme has two parts, the substrate and the active site. Okay, so the substrate is the substance or the reactant that binds to an enzyme. So whenever you are having a chemical reaction and you want something, you want basically a starting molecule to turn into a finalized or finished molecule. This starting molecule is known as a substrate. Is known as a reactant and a substrate, I guess. So basically, a reactant or a substrate is the starting ingredient that you are trying to use in order to create a finalized molecule or a finalized compound during a chemical reaction. And this substrate will bind to an enzyme so that it turns into a product much faster. So I added an example of reactants and products just so it's more visually, clear. So here we have a chemical reaction which involves the formation of H2O, which is water. As you can see, the reactants are H2 and O2. So we have two atoms, one atom of hydrogen, another atom of oxygen, combining to form water. Okay, so basically the reactants in this example are the hydrogen and the oxygen. And the products, or the product that is being made by the end of this chemical reaction, so this whole thing is a chemical reaction, is water. Now, if enzymes were not involved in this chemical reaction, then it would take forever to turn hydrogen and oxygen, to have them combine, to have them form a covalent bond and then make water. It would take too long. So to speed things up, enzymes help hydrogen and oxygen to combine to form a covalent bond and form water. And in this scenario, the substrates, okay, the things that are going to be binding to the enzyme is going to be the hydrogen and the oxygen. Okay, so these are the substrates in this example. And substrates will always bind to a specific place or location in an enzyme known as the active site. So the active site is the area on the enzyme where this substrate will bind. So you have the entire enzyme, and there's a very specific place on the enzyme that has a very specific shape. This is known as an active site, and this is where the substrates will bind. Now, whenever anything happens that disrupts homeostasis in the body, this can affect how these enzymes work. Now, we already know that enzymes are proteins, and the issue with proteins is that they are super sensitive to any changes in the environment around them. So in the body, if the temperature gets too hot or pH gets too acidic or too basic, what that's going to do is that's going to change the shape of this enzyme. It's going to make it, we call it denatured. Okay, it's going to ruin its shape. And if the shape of this protein, if the shape of this enzyme is ruined, then it can no longer do its job. Structure and function are extremely important. So here we have an example of 3 different enzymes that are found in our body. Pepsin, which is found in our stomach, and it's responsible for breaking down a specific kind of food. Salivary amylase, which is found in our mouth, and it also breaks down certain kinds of food. And then alkaline phosphatase, which is found in many different parts of our body, including our blood. So these 3 different enzymes are responsible for maintaining homeostasis in different ways. They act on different things in the body. And you'll notice that each one functions at a very specific pH. So this is the scale on the bottom here is actually showing pH. Once that thing moves out of the way, you'll be able to see it. But the idea here is that each one of these enzymes is functioning at a very specific pH. So pepsin functions best at a pH of around 1 point something. And that makes a lot of sense because pepsin is located in our stomach. So it loves an acidic environment because that's what the stomach's environment is all about. Salivary amylase functions around 6 point something. So you'll notice that is kind of neutral. Okay, it's slightly acidic, which is actually what the environment in our mouth is like. And so on and so forth. So the idea here is that each one of these enzymes functions best or optimally, that's like the fancy scientific word, at a specific pH. And if that is changed for some reason, then the enzyme will no longer be able to function. So to summarize, what I'm trying to say here is that the enzymes, they are super sensitive to changes in temperature and pH. If the ideal or optimal temperature and pH changes for that specific enzyme in the body, it will no longer be able to do its job because its shape, its structure will be ruined. Now, like we said, an enzyme structure is very specific and it has, several parts that we've already discussed. The substrate, which binds to this area known as the active site. And by the end of the entire chemical reaction, the enzyme will produce the product. And if you remember, the product is basically the final thing that is made at the end of the chemical reaction. So in this specific example, the product was water. So the way that enzymes work, we often describe it using a lock and key model. So the lock and key model helps us explain how enzymes do their job. So the whole process begins, like we said, with the substrates or the reactants binding to a specific location on the enzymes known as an active site. These reactants are brought together and the enzyme brings them together at a much faster rate than they would on their own. The substrates are combined, chemical bonds form between them, and then sorry about that. And then finally what happens is that we end up with a final product being formed. Okay? So enzymes have the ability, luckily, to be recycled. After they do their job, after they form these products, they end up, they're kind of like just lying around in the body and they can actually be recycled. They can be reused over and over and over again, as long as their structure isn't ruined, like we discussed in the previous slides. And that is about it. So just to quickly summarize what we talked about, enzymes are biological catalysts. They are found in the human body. They are found in the bodies of all living things. Enzymes have very specific shapes. Their structure is very related to their function. And a main area or a very important part of enzymes is this specific location known as an active site. The substrates or the reactants bind to an active site of an enzyme, and it brings them together so that at the end, a product is formed. This whole thing that we see in front of us here is known as a lock and key model, which is kind of like a model that we use to explain how enzymes work. And to summarize what enzymes do, like how do they work, what are they doing in the body, enzymes speed up chemical reactions.